Okay, so welcome back. Now we are going to look at some more types of azeotropic distillations or extractive distillations. Recall from the previous lectures, rather I should say, how do you generate residue curve maps? How do you generate approximate residue curve maps? How do I use them for deciding about sequence of distillation columns? Now we are going to continue in the same way. We are going to look at now one more type of distillation. So now it is going to be azeotropic distillation, meaning the entrainer is light, but now it is heterogeneous distillation. Heterogeneous azeotropic. Heterogeneous means it is going to form two phases at least in some part of the ternary mixture. So if I have a mixture of A and B, they form azeotrope. We are going to add C as an entrainer. The entrainer added is light. That means it is going to go to the top of the distillation column along with one of the components, but it is going to form two phases at least somewhere in the distillation column, or once you condense, two liquid phases. So like an aqueous phase and an organic phase. And many times azeotropes are formed when water is there, or sometimes we could add water to break the azeotrope, in the sense to generate the two liquid phases, and we will see how this generation of two liquid phases helps us to separate the two components A and B which have actually formed an azeotrope. So many times one of the components is water, either A is water or B is water, means more volatile is water or less volatile is water, and then we add a third component which forms another azeotrope with water, and then it goes to the top of the distillation column because it's a light entrainer. We call it as an azeotropic distillation, but at the top it splits into two phases when we condense into an organic phase and at an aqueous phase. Okay, so let's take some examples. Let's see how these these can be done. So one of the examples is pyridine and water. So when you make pyridine, you generate pyridine using formaldehyde, acetaldehyde and ammonia, and in that reaction a lot of water is also generated. So you, at the end of the outside of or what is coming out of the reactor is pyridine, these unreacted components, and water that is formed. Of course there are some impurities like picolines and all that also form. Let's just take an example that we have only pyridine and water, but then if you have to purify, I have to separate pyridine and water. But now pyridine water form a minimum boiling azeotrope at 77 mole percent water at 95 degrees centigrade. But I can add toluene as an entrainer. Now you see, we are calling it as an entrainer, we are calling it as a light entrainer. It has a boiling point not lesser than 100, but it has a boiling point of 111 degrees centigrade. So it's strictly speaking not as a light entrainer, but you see this toluene water forms a minimum boiling azeotrope at 85 degrees centigrade. So this is the lightest, this is the most volatile of the mixture. So 115 is pyridine, water is 100, they form azeotrope at 95, but this toluene water azeotrope is at an even lower temperature, so it has the most volatility. Therefore this toluene water azeotrope will go to the top of the column, therefore we call it as a light entrainer. But toluene and water, of course there's one more problem here, pyridine and toluene form a low boiling azeotrope at 110 degrees centigrade. Okay, now coming back, now this toluene and water is the lightest, in the sense it's most volatile, the temperature is the lowest. Now if it goes to the top of the distillation column, it will split into two phases, aqueous phase and organic phase. Aqueous phase essentially containing water, organic phase essentially containing toluene. Okay, we are going to use that to our advantage. So what I want you to do is to draw residue curve map, propose a process flow diagram, indicate the feed distillate bottom composition on the residue curve map, and calculate quantities and compositions of all streams per 100 kilogram per 100 kilomoles of the feed mixture. And feed mixture is, let's say, feed contains 10 mole percent of pyridine. Okay, first I want you to draw an approximate RCM. I want you to draw an RCM for yourself. Pause the video, do it for yourself. Only then you should go forward. Only then you should go forward. Okay, I want you to draw a residue curve map like this: pyridine, water and toluene. Pyridine here, water here and toluene here. Remember, water toluene form a two-phase mixture, heterogeneous region. So in a triangular diagram we are showing that two-phase region in this manner. So this is the region and these are called as tie lines. Do you remember that from your undergraduate course? We have tie lines, we have a plate point here, and inside this we have two phases: aqueous phase, organic phase. Their compositions that are in equilibrium in the two phases are given by the tie lines. So aqueous phase will have this composition, organic phase will have this composition in equilibrium, but somewhere here it forms a homogeneous single liquid phase. So now, in addition to the residue curve maps, the location of this two-phase curve, two-phase envelope, two-phase region is also very important. Okay, so draw this residue curve map, approximate residue curve maps, including the two-phase region. Okay, pause the video and only when you're done, continue. Okay, so here is how the residue curve map will look like. Pyridine, pure component, water, pure component is here, toluene, toluene water form an azeotrope here. This is a minimum boiling azeotrope. So the residue curves are all pointing away from the water, from the azeotrope towards water and towards toluene. Pyridine water again form a minimum boiling azeotrope here. The residue curves are pointing away. Pyridine toluene, no azeotrope. So residue curves will only point, pyridine water, pyridine toluene, there's another azeotrope, minimum boiling azeotrope here, 110. So the residue curves will point away from this azeotropic point towards the pyridine axis. And you see, this is the two-phase region. And these are all the tie lines. One tie line, second tie line, third tie line, fourth, fifth, so on. These are all the tie lines. Inside this we have two phases. Outside this we have single phase. So if anywhere mixture is here, let's say for example mixture is here, it's split into two compositions or two mixtures, two phases, one aqueous phase having this composition and an organic phase having this composition. If my composition is like this, it will split into two phases, one aqueous phase containing essentially water and pyridine, very little toluene, another organic phase containing toluene and pyridine with little quantity of water. So that's the meaning of the tie line, and that's how we could, if the composition is anywhere, we could get a compositions of the liquid phases, aqueous phase and organic phase, which are governed by this tie line. Recall, this is from your undergraduate class, undergraduate course. Okay. Now with this residue curve map, now you tell me how we are going to do the distillation. What is our feed? Feed is 10 mole percent pyridine. That means this is our feed here. Our feed is here. Now how are we going to do? How are we going to do? Pause, think about it yourself. Suppose I add toluene. Say I add toluene. My composition will be somewhere along these lines, along this line. Suppose I add too little toluene, I would be somewhere here. Now if it will split into two phases because it is inside the two-phase region. One phase I will get essentially pure pyridine and water. Second phase I will get a mixture of pyridine, toluene and little amount of water. If I add too little toluene, no sorry, if I add too much toluene, maybe I would be somewhere here. Now my tie line is going to be something like this. I will get essentially one phase containing almost water, very little pyridine, and the second phase containing almost pyridine and toluene, very little water. Okay, but we would like to do this using distillation because if I get pyridine and water, even if I get a essentially pure pyridine and water mixture, pyridine and water form azeotrope. Sorry, if I if I add two, if I get add too much toluene, I'm here I split into two phases, essentially pure pyridine and water, pure water, very little pyridine, and a pyridine and toluene mixture, very little amount of water here. But that also I can't separate because pyridine and toluene form an azeotrope, right? They form azeotrope, so I can't separate them easily. So I need distillation, right? So one of the ways, think about it, how you would do using distillation. Okay, one of the ways, here is the answer. Don't look at the answer till you have attempted it yourself. You may not get the answer, but try it yourself. Okay, so suppose I add toluene such that I end up with my mixture here, but I don't do separation, I do distillation. At the top I will get what? At the top what I will get? At the top, look at the residue curves. Residue curves, this is one distillation boundary. This is another distillation boundary. I have one distillation region, I have another distillation region, I have third distillation region. Look at the residue curve maps. If you want to draw residue curve maps, okay, your residue curve maps will look something like this. Right, we are starting with the lowest temperature. Recall how we started with the residue curves. Your residue curves will look something like this. This would be one region. We started with the lowest temperature. We wrote order of temperatures. This would be another residue curve. This would be the region. And if I am on the other side, this would be the residue curve. You realize? So there is a distillation boundary here. Let me draw this properly. There is a distillation boundary here. There is another distillation boundary here. Actually these are curved, so these actual boundaries are curved. Approximate distillation, approximate residue curve map if you are drawing, you would have not predicted the curvature, you would have predicted the boundary like this. But that's okay, that's fine for now. So now if I add to this feed, if I add toluene, I would be, the mixture composition would be somewhere along the line joining the feed and the pure toluene. Let's say I end up here. Now if I do distillation, now look at the residue curves. Residue curves are pointing towards the pyridine, which means I will get at the bottom, I will get pyridine. At the top, after condensation, I would get a mixture, essentially an azeotropic mixture of water and toluene. So that will split into two phases, one aqueous phase and an organic phase. Organic phase will be essentially, now it will be pure toluene. You see, toluene with very little water, and I will get water. You see the two-phase envelope. The two-phase envelope is practically touching this axis here. It is practically touching the vertical line. That means it is going to contain very little toluene. The two-phase region is practically touching the side of the triangle. That means it is going to contain very little water, right? So if I have a mixture of pyridine and water, let's say 100 moles, 10 moles pyridine, 90 moles water. I add toluene. Suppose I add 100 moles of toluene. Then I end up with a mixture here, let's say 100 moles. Then I will get water, essentially 90 moles coming out, and organic 100 moles toluene, which I can recycle back. And actually we don't have to do this I don't have to add a separate mixer and mix the toluene and pyridine and water, because they are going to form anyway heterogeneous phases. They are going to form two phases. We can do this in a distillation column itself. So decanter would be here, water would come out, and the organic phase toluene I can recycle. So the toluene keeps on going down, keeps on going up, keeps on getting refluxed. So the toluene keeps on moving around like this. And in this top part of the distillation column, we would have a two-phase mixture. Bottom part, we were essentially pyridine and water, and then pyridine is getting enriched in the bottom. Water is getting stripped from the pyridine because of the toluene, because of the azeotrope that the toluene is forming. Okay, this is how you could do it. Right. Another way is to look at, okay, I could have a pre-concentrating column. So this is our azeotropic column. This is what I showed you here. But I can also have a pre-concentration column that I will enrich myself into more pyridine. I could go from here all the way up to azeotrope, right? This is the azeotrope. So I could do this going from this feed to the azeotropic composition. I could do this in a pre-concentration column, right? And then this mixture, the azeotropic mixture of pyridine and water, now we can feed to toluene, right? So we would be somewhere along the line joining this azeotrope and toluene, and perhaps I add equimolar mixture of the azeotrope and toluene. We end up somewhere here. Now of course I can do the distillation. I'll get pure pyridine at the bottom. I will get azeotrope at the of toluene and water at the top of the column. Pyridine at the bottom and azeotrope of toluene and water at the top. Okay, so this could be pre-concentration, could be one column, say here. So then I am getting pure water at the bottom. I have got an azeotrope of pyridine and water at the top. Now I keep on recycling the toluene. Toluene comes from the reflux. Toluene water azeotrope goes to the top, which separates into two phases. The organic phase containing toluene, which is recycled. Aqueous phase containing pure water, essentially, goes out. And bottom of this distillation column, I get pyridine. So if you see this as a whole plant, we could have a pre-concentration column, or we need not have a pre-concentration column. I have a feed containing pyridine and water. I have separated out water. I have separated out pyridine. Toluene, I keep on recycling from the decanter to the column. It keeps on distilling along with the water. So now you see why it is called as a light entrainer. Toluene per se boiling point is higher than water, but light because toluene water forms an azeotrope which is lightest, which has the lowest volatility amongst these. So it's called as a light entrainer. It's called as a azeotropic distillation. And heterogeneous because toluene and water form two phases. So one more point that we have to be careful about, that in this column we can have two phases being generated inside the column, because toluene in this region, somewhere in this column, we could be in this envelope, we could be below this envelope, and we could have a two phases forming inside this column itself. So I would better have a tray column here, because if I have a packed column, then I don't have a good contact between the vapor and the liquid, two liquid phases. Whereas if I have a tray column, then even if two liquids form on the tray column, the vapor could bubble through the tray, bubble through the pooled collected liquid, and then go to the allow the vapor liquid equilibrium to be reached. So the choice of packed column versus plate column is also dictated by whether we are going to have two phases inside the column. So if we are having two phases inside the column, we better use a tray column so that we can have a good mixing between the vapor and both the liquids, so that we can get a nice good vapor liquid equilibrium between the two liquids and the vapor. Okay, another example, acetic acid and water. So acetic acid and water, they do not form azeotrope, but their alpha relative volatility is very close to 1. So sometimes butyl acetate, isobutyl acetate, boiling point 116.4 is used as an entrainer. Now you see 116.1, it is more than 100, it is very close to 118. So boiling point wise it is not a light, but butyl acetate and water form an azeotrope which is having a boiling point of 87.7 degrees centigrade. So this butyl acetate and water form a azeotrope which is having a temperature lower than 100. So we call it as a light entrainer. So we call it as a heterogeneous distillation, heterogeneous azeotropic distillation, because butyl acetate and water form two phases. Solubility of butyl acetate in water is about 0.7%, and solubility of water in butyl acetate is about 8% mole percent. Right? When acetic acid concentration is about 30%, the mixture forms a single homogeneous phase. So what I want you to do is draw RCM, residue curve map, including the two phase region. Again, I want you to draw this acetic acid at the top of the vertex, water and butyl acetate on the two sides of the triangle, bottom of the triangle. Okay, do this. I want you to do it yourself, and only when you have done it yourself, go forward. And as usual, indicate the distillation columns on the RCM and calculate the compositions and the quantities of all streams. That's as usual. Pause, do it for yourself. Pause the video, do it for yourself. Don't go forward unless you have done it for yourself. Don't go forward unless you have done it for yourself. Okay, here is how the residue curves are going to look like. Acetic acid, top vertex, water here, isobutyl acetate here. This is 116.4, this water is 100, acetic acid 118, acetic acid, water, they do not form an azeotrope, but water butyl acetate form an azeotrope. It's a minimum boiling azeotrope. The residue curves are pointing away from the azeotrope towards these individual. Water acetic acid is less volatile, they form, the residue curves are like this. Residue curves will look something like this. If you are on this side, it will go like this. If you are on this side, it will go like this. And of course, isobutyl acetate, acetic acid, the residue curves on the binary of acetic acid and isobutyl acetate is towards the acetic acid, 118. Right? So this is how residue curves will look like. This is the two phase region. Right? Water aqueous phase with very little butyl acetate with maybe 8 mole% of water. Right? This is how the two phase. Anywhere below this curve, I can create out of these residue curves, we form a two phases, and these are the tie lines. This is one tie line, this is second tie line. The dotted line is the two phase envelope. Below this dotted line we have two phase mixture. Above the dotted line we have a single phase mixture. Right? That's what the meaning of these sentences. Okay, now let's say what is our feed? Well, feed, let's say, calculate the per 100 kilomoles of feed mixture containing 30% acetic acid. Right, let's calculate. Propose a process flow diagram. Can you pause the video, do it for yourself? Now feed is here 0.5, either 50% or 30%, so don't worry about this. Don't worry about this. Say feed is here. Okay. Now suppose I add butyl acetate. I would be somewhere along this line joining the butyl acetate and water, joining the butyl acetate and the feed. So let's say if I add something like this so that I get a mixture, this is my feed to the distillation column. Now if I distill, look at the residue curves, they are all pointing towards acetic acid. So if I do a distillation, I will get acetic acid at the bottom. Top would be essentially butyl acetate and water azeotrope. That will split into two phases, one here and one here. Actually, I don't need to worry about too much about where my feed is. Even if I add, let's say I join this line, now do this properly, you should be able to do it properly. If I add too little feed, maybe I will be here. And now if I distill, I will get the, at the top of the column, I will get water and butyl acetate containing about 80% water, 20% butyl acetate. But it's going to split into two phases. One phase here, one phase here. There's no distillation boundary, so there's no worry. Even if I add too much butyl acetate, maybe I will be here. I would still be able to distill out. I will get distill out. The acetic acid will come to the bottom. Top, I will have maybe 75% butyl acetate, 25% water. That's still going to split into two phases, one here and one here. Okay. So I don't have to ensure that I reach this azeotrope point. I could add, I could work with this or this or this. Even if your feed is 50% acetic acid, let's say even if this is your feed, right? Now I could draw a line joining this feed along with butyl acetate. Maybe I will be here, I will be in a homogeneous region, but maybe I add equal amounts of feed and I add equal amount of butyl acetate. So I am on a line equidistant from both the points, so I am maybe around here. That is my combined feed. Now I distill out, I get this acetic acid at the bottom. I would get this 60-40 mixture of butyl acetate water at the top of the column, which will split into two phases. So now draw the process flow diagram and see what you can recycle, how you can recycle. Okay, so one possibility is this. This is our feed mixture, 30% acetic acid, 70% water. I send this to an azeotropic column. This is my azeotropic column, heterogeneous azeotropic column. I distill it out. I get pure acetic acid at the bottom. Top, I have essentially butyl acetate and water, two-phase region. I end up somewhere here, either here or here or here or here. Anywhere I want to end up, that's okay. So in this particular case here, we are having about 26% water, 74% isobutyl acetate. That means we are here. 26% water, 74% butyl acetate and water somewhere here. Now it's going to split into two phases. Organic phase here, which is essentially butyl acetate containing about 8% water. This can go back to the column. And an aqueous phase, let me draw with a blue, which is containing essentially water but very little butyl acetate. You see how much butyl acetate is soluble? 0.7%. butyl acetate is soluble in water, 0.7%, you recall. So this stream is essentially 99.3% water, 0.7% butyl acetate. Now 0.7% butyl acetate, if this water is to go to ETP, it still corresponds to 7000 COD, 7000 ppm of 7000 ppm of isobutyl acetate, which will correspond to about maybe 20,000 COD. So I still need a stripping column, which will, so essentially I still have, I have ended up here, the water phase is here. So I will still strip it out, I will get water at the bottom, I will strip out this azeotrope at the top. So you see the residue curves are pointing here. So in the second column, I still have 99.3% water, 0.7% butyl acetate, but butyl acetate is costly, I don't want to lose that. So if I distill this mixture, I will still get the azeotrope at the top and pure water at the bottom. So you see, I'm having pure water at the bottom, I have an azeotrope at the top, 2178. Right, 21. This is in terms of 2178 is in terms of weight percent. In terms of mole percent, what was it? In terms of mole percent, it was 36 mole percent isobutyl acetate. Right, 36 mole percent isobutyl acetate. Right, so weight percent, mole percent, just take care. So now this azeotrope, I send it to condenser. It condenses into two phases, isobutyl acetate, which I recycle. Water goes to this column. So you see, now this is my whole plant. I have separated it into water, acetic acid has separated into two pure streams, essentially pure acetic acid, essentially pure water. Whatever butyl acetate is there is just recycling in this loop, it is just recycling in this loop, the organic phase and the aqueous phase. It is the butyl acetate just keeps on recycling here. Small amount of butyl acetate is lost from condenser and so on. I have a makeup quantity of isobutyl acetate which can straight away go to the condenser and decanter. Okay, so although acetic acid water do not form an azeotrope, sometimes it helps to use separate them easily using an entrainer. Okay. Pause this video, understand for yourself. Pause, don't go forward till you have understood.